Welcome, we're group 7 from section 1, and this is our last lab practice called diffusivity of a mosquito repellent. We've divided our presentation in several sections, which are introduction, proposed methodology, lab methodology, results, analysis, conclusions, and finally our references. The importance of the mosquito repellent is to help protect from mosquitoes that spread different diseases such as malaria, dengue, chikungunya, and yellow fever. It can be used on top of the skin, on the clothes, or for the ambience in general. And the higher the diffusivity the repellent has, the most effective it is. For this lab practice, our group used bay leaves as a natural repellent, and bay leaves extract has the secondary metabolic compounds such as terpenoid, known as a repellent for mosquitoes naturally. And we made this repellent from 2 cups of water and 10 grams of bay leaves. What will be analyzed in this lab practice is the diffusivity of our mosquito repellent, and the diffusivity is defined as the ability of a substance to move through a medium or material. In diffusion, particles move from a region of high concentration to a region of low concentration due to thermal energy. To calculate diffusion, it is usually used the Fick's law, which is shown on the left, and on the right it is shown its approximation. However, neither of these can be used in our lab methodology because we can't calculate the diffusive flux. As the fixed law can be used in our case, we'll be using the Sorot's equation shown here. However, as our system is in a steady state and there is no temperature change inside the control volume, and the thermodynamic coefficient is neglected due to there is no thermal exchange between species, we have to eliminate these three terms and we obtain this simplification of the equation. With everything said in the introduction, our objective in this lab practice is to determine how temperature affects the diffusivity of our repellent. Now we will show our proposed methodology. The steps proposed in our methodology are the following. First, we need to use two containers communicating through a tube, and then weight the repellent and put it in one of the containers. Then, using a chronometer, time when the odor of the substance begins to be perceived on the other side of the tube. And with this time, we can calculate the diffusive flux. And thanks to the diffusive flux and the fixed equation, we can obtain D, the diffusivity. And finally, we can repeat this process for different temperatures and analyze how the temperature can affect the diffusivity. Now we will show the methodology that was used in the lab. In the lab, we started with 4 containers, each of them marked with a different temperature, which were 10, 30, 60, and 90 Celsius. Then to each container, they were added 30 milliliters of water at the respective temperature. Then to our repellent, red dye was added, and then distance in each container was marked, and this was our Y. To each container, 3 millimeters of repellent were added, and samples were taken out of the distance marked as Y every minute for 4 minutes from each container. Then, thanks to the UVVs, the concentrations were analyzed, and with these concentrations and the Soret's equation, we were able to obtain the diffusivity.
And now we are going to present the results. First, the UV-vis spectra of the samples obtained at each time for each temperature were obtained. Here we can observe the spectra of the samples at 10 degrees Celsius and 30 degrees Celsius in the range of 600 to 800 nanometers. In this case, the maximum absorbance was used to calculate the repellent concentration. This was obtained at 610 nanometers. In that wavelength, There is a local maximum that can be evidenced with a slight band in all the spectrums. In addition, the decrease in absorbance over time can be seen. The same thing was done for the UV spectra at 60 degrees Celsius and 90 degrees Celsius, where we can evidence the same local maximum mentioned above at 610 nanometers. However, here an increase in absorbance with time can be seen. Once the wavelength of maximum absorbance was selected, the concentration of each sample at each temperature was calculated using Lambert-Beer's law. To do this, the molar extinction coefficient of water was necessary, which was the substance in the highest proportion in the repellent. It was found that this was 1.349212 around 600 nanometers. So it was selected, taking into account that the difference with the selected wavelength was only of 10 nanometers, and the extinction coefficient of water does not present a great variation in the visible spectrum. On the other hand, the standard length of a cell was used, which was 1 centimeter. After calculating the concentration at each time, it was possible to determine the diffusivity with the simplification of the Sauer equation that can be seen here, in which the difference in concentration is between time 4 and time 1, just as time and distance corresponds to the height of the mark made, that was 1.7 centimeters. For the water at 10 degrees Celsius, we obtain a diffusivity of 0.00133023. The same was done for all temperatures. In the case of water at 30 degrees Celsius, here we can see the results of the concentration over time and the diffusivity, which was minus 0.00015455. For water at 60 degrees Celsius, a diffusivity of 0.00000903 was obtained, despite the fact that the concentration increased over time. The same thing happens with water at 90 degrees Celsius. The concentration increased over time, but a diffusivity of 0.00000343 was obtained. Finally, we can observe the graphs of diffusivity as a function of temperature and as a function of the difference in concentrations with time. In terms of temperature, an exponential decrease in diffusivity can be seen as the temperature increases, and with concentration, we can see something similar except from an outdated value at the end. This will be explained in a moment.
Now we will move on into the analysis. Through the graphs regarding the wavelength versus the absorbance, it is possible to see how there is a peak at 610 nanometers. This means that at that wavelength, light was absorbed from the repellent. This means that at that point, the quantity of absorbed light depended on the concentration of the solution. This is demonstrated by the fact that by bigger absorbance, there is bigger concentration. It is worthy to mention that in future experiments the wavelength range could be bigger, probably from 100 nanometers to 800 nanometers. This could show the presence of terpenoid and eugenol in the repellent, since they are present in bay leaves and are mostly seen around 200 nanometers. By understanding the peak that was shown approximately in 610 nanometers, it was possible to get the concentrations and the diffusivity coefficients in the different temperatures. This was done using the simplified Surrey equation. As was explained at the methodology, the velocity term of the equation is not considered because the system is at a steady state. The thermic diffusivity is also not considered because there is not a temperature gradient inside the beaker. And the gamma coefficient is not considered because there is not a thermodynamic relationship between the components in the system. It is possible to see throughout the results regarding diffusivity and temperature how it is decreasing exponentially. In typical experiments, it has been shown how diffusivity increases while temperature increases. This is because the repellent is trying to enter an equilibrium state with air, so the molecules of the repellent are moving in the direction of where the air is. While the temperature is increased, there will be an increase in the kinetic energy of the molecules, and in result, they will move faster towards equilibrium within the medium they're in. This results in bigger diffusivity. In this case, the repellent used was made with bay leaves. This herb has its main components of eugenol, cineol, and terpenoids. For example, eugenol and the terpene have shown to have low solubility in water and are non-polar. This may result in the fact that the main components of bay leaves are hydrophobic and they cannot interact with the hydrogen bonds from the water molecules due to the low solubility they have with water. In return, water creates an organized structure within itself and creates even more organized movement. This doesn't allow diffusivity to increase correctly. This explains why at the moment of increasing the temperature, the water will still reject the main components of bay leaves and in return the diffusivity will decrease. Now, in terms of the concentration and diffusivity, it can be seen how at bigger concentrations of repellent, less diffusivity, and then it begins to change. At bigger diffusivity, more concentration. This can be compared to typical behavior between methanol and water combined. At the beginning, while it is added repellent to water, the diffusivity will decrease because the water molecules are organizing themselves and there's no much movement for the repellent. Then it comes a point where the diffusivity enters its limit and now the repellent added will be more than water and the concentration will rise. There is a point in the experiment where, meanwhile the diffusivity grows, the concentrations returns, and it can be rooted in experimental errors. It is possible that in that temperature, the selected sample went beyond the 1.7 centimeter mark, and in return, the concentration calculated did not match the natural behavior of the diffusivity.
When we compare our proposed methodology to the one used in the lab, we can see some differences. The first one is that in the proposed methodology, the fixed law could have been used thanks to knowing how to calculate the diffusive flux by knowing the area of the tube. However, this couldn't have been used in the lab methodology, and this is why we use the Sorentz equation. There is less error for the proposed methodology because we had some difficulties in the procedure for the lab methodology because when taking out the samples, the liquid could have been agitated, affecting the results. The materials used in the proposed methodology are simpler because there's only two containers and one tube. But in the lab methodology, we did use an equipment called the spectrophotometer UVPs.
Finally, we will move on into the conclusions. In conclusion, it can be seen how due to the components of bay leaves, such as eugenol and terpene, there will be a decrease in diffusivity while the temperature increases. The main components of bay leaves are hydrophobic, and this results in water rejecting the bay leaves and restricting movement in the solution. This decreases the diffusivity of the repellent. Additionally, there is also a natural oscillatory behavior regarding the concentration and the diffusivity. The last set of data does not look natural because of experimental errors. Additionally, there was a big amount of experimental errors. At the beginning, it was a research how the main components of the bay leaves were hydrophobic. At the moment of the experiment, it was possible that the data collection was not diligently done. The exact place where it was supposed to obtain the solution may not have been followed. And probably the bay leaves also had other components in its packaging and more. In regards to the data analyzed, it is suggested that in future experiments in the absorbance analysis there is a bigger range regarding the wavelength. Finally, here are the references used. Thank you very much.